Hey folks, so my name's Dennis Mishler, Dr. Dennis Mishler. I'm a professor here at the University of Texas in Austin in the United States, and I've been part of iGEM for a number of years now. I've had a number of teams. I've been a judge for several years, and I've been a member of the measurement committee for a couple of years. So today we're going to talk about transformation, conjugation, and DNA sequencing. The first two of these are techniques to genetically modify bacteria. And the last one is a way to look at your DNA constructs to figure out if you have the right sequence or not after you've done all your cloning efforts. So let's go ahead and get started here. So transformations. So why does it matter? Well, like I said, it's a method for genetically modifying your bacteria. And in particular, we're going to focus on genetically modifying E. coli. So I'm not going to talk about transformations for other organisms in this section, only E. coli really. So whatever I talk about, this is applicable to E. coli generally, right? So you got to get your DNA inside the cells. You're going to do a transformation procedure. Sounds good. Your end result should be these plates here. Okay, you should be getting colonies on plates, bacteria growing on the plates. And you can use transformation to transform different types of DNA into bacteria. You could use ligation DNA. So if you've done a digestion ligation reaction, you can take that DNA and try to put it into bacteria. If you've done a Gibson or Golden Gate assembly reaction, you can take that DNA, try to put it into bacteria. If you already have a construct that you've acquired from another lab or from somebody else, you could take mini prep or plasmid prep DNA and transform that into bacteria to make more of it. That's all good. And the end result, right, is going to be that you have a genetically modified organism. Okay. So what's needed for a transformation? Well, first off, we're going to need some sort of competent cell. Competent just means it is competent or ready to be transformed. Okay. And there are many different strains you could use for different purposes, and there are many different procedures that you can use to make competent cells. One example, let's say you're trying to create a protein expressing cell line. Well, you might want to use a BL21DE3 cell for that. But if you want to make a plasmid, a cell line that focuses on plasmids, you might make a different type. You might use a different type of competent cell. So one that's more attuned to making lots of plasmid as opposed to making lots of protein. Okay. There's also going to be different sources of DNA, like I mentioned on the last page. You could use ligation DNA, assembly DNA, mini prep DNA, something along those lines. And those different sources of DNA are going to give you different success fail rates with higher or lower. One second. I've just been told I'm sharing something a little different. One second here, guys. Let me see if I can figure out what's happened here. All right, I've just changed the sharing settings. Does this fix the problem? Do you guys just now see the presentation? No. Uh-oh. Okay, give me a second. Sorry about this, folks. Thank you, though, for telling me. One second. How about that? Did that help?
No, you're still sharing the presenter view, not the presentation.
I'm sorry about this, guys. Let's try this. Did that work?
Yes.
Okay, great. My apologies, everybody, for taking care of that. Yeah, thank you for the different suggestions there. Okay, so maybe we can get Okay. All right. So DNA sources, like I said, different DNA sources will give you different rates of success. So if you have a mini prep DNA, for example, which is going to be a better quality source for plasmid, you're going to get a lot more transformation success than if you're using ligated DNA or an assembly DNA, which is going to have not just all perfectly made plasmids, but also plasmids that are nicked or incomplete sequences. So you have to take that into account when you're doing your transformations. The means of transformation, this is how we're going to get the DNA inside the cells. I'm going to talk about that on the next slide. Recovery, so after you've transformed your cells, you got to give them some time to recover, and we'll talk about this in a little bit. And then once the cells have recovered from the transformation, you've got to put it onto selective media. So normally this is going to be like an antibiotic on a plate. And from this, you want to ideally get single colonies growing up. And then finally, you need to do some sort of quality control. Normally this is going to be DNA sequencing to make sure that your construct was actually correctly made. Not just that it looks good, but that the sequence is actually what you expected. So transformation. So this is how we're going to get our DNA inside the cells. And there's two main ways you can do this. One is going to be a chemical transformation. And the second one is going to be an electroporation transformation. So you can see up here chemicals A on the top and electroporations B on the bottom. And we'll go through each of these real quick. So chemical transformation, you can see the first step, it's pretty, actually A and B are pretty similar in their steps. So the first step here for chemical, you're going to take your chemically competent cells, which you've made previously using a procedure to make them. You're going to take those cells and you're going to just add the DNA to them. You're going to let them sit for a while. And then the very important step in chemical transformations is you have to do a heat shock. You're going to get a water bath, warm it up, and then expose your cells to that for a little bit of time. In this particular procedure that's written out here, they went with 42 degrees Celsius for 30 seconds, but that can change a little, that can be varied. So depending on your procedure, you might use a slightly different setting, and that's okay. But after the heat shock's over, you're going to cool your cells back down. And then from here, you would go back to the recovery. So you'd start the recovery process to let the cells grow and start to express your DNA sequence. Now, as I mentioned, to do chemical transformation, you need these chemically competent cells. And you would have to make these in advance, all right? And that's a relatively simple procedure, and I'll show you a link in a little bit that can tell you how to do that. But you have to do that in advance. B, electroporation is a very similar progression. You first take your electrocompetent cells, which you've previously made, you add DNA to them, let it incubate for a little bit, and then here's where things get different. For electroporation, you have to actually electroporate or essentially send current through your cells. So what you're going to do is you're going to transfer the cells into this electroporation cuvette, and this is then going to go into a machine. You're going to press a button, it might say pulse or shock or something like that, and a little current's going to go through the cell mixture. And when that current goes through, it'll allow the cells to open up a little bit to take in the DNA. And then after that, you're going to cool the cells down maybe for a little bit, and then again, you're going to go to the recovery step. But these two steps here, the heat shock versus electroporation, that is the main difference in these processes, all right? Both of them use cells that you've previously made competent, and then both of the processes, once they're done, go to the recovery step, all right? This image here was grabbed from the Thermo Fisher website, and they actually have a really nice page that goes into the protocols for each of these types of transformation processes, as well as some of the steps you need to do to make competent cells, and they explain some of the basic concepts behind it. So if you're curious, you can definitely read more there and realize that they're going to be referring to their type of competent cells, the cells that they would normally sell you. Okay? But it's a very nice site to get some information. So once you've done your transformation, whether it was electroporation or heat shock, you're going to need to let your cells grow for a little bit. And the main reason for this is that on your plasmid, you should have a selectable marker, such as an antibiotic resistance gene. And that gene needs to be transcribed and translated for a while before you put your cells onto a plate with the antibiotic. And this is really important because if you have an antibiotic that stops all translation, if your cells have not already translated the gene that gives them resistance to that antibiotic, well, they're never going to, right? The antibiotic is present, it's going to halt all translation. So it's really important that you let your cells recover for a little bit before you plate them. Now, this varies. The time varies quite a bit depending on the selectable marker. In general, ampicillin or carbenicillin which are both beta-lactams, you can just do a small, short recovery for those, like 15 minutes, and then you can plate and you're good to go. For most other antibiotics, though, you really want to do a recovery for about an hour, sometimes more. But you just want to read about your own antibiotic and what to do based upon your experiment. After the recovery is done, you're going to go ahead and plate your cells, your transformation mix, on selected media. And your goal from this is to have successfully transformed cells that are growing on the plate, and ideally as isolated colonies. Okay, so this circled one down here, for example, would be is an isolated colony. It's isolated from the other colonies, so it's further away, and then it's a colony of cells. And a colony of cells, such as this one right here by itself, or the one that's circled, it originates from a single cell. A single cell, when we were plating out the bacteria on the plate, a single cell went there and just started growing, and it made more and more of itself and became a cluster of cells that we could just call a colony. And so every cell within that colony should be genetically clonal. They should be the same. Now, realistically, that's never the case. There's always a few mutations here and there. But with regards to your plasmid, you would expect all the plasmid sequences within that colony to be identical, all right? But to confirm that, we'd really need to go on to quality control. And so once your transformation is done, you would pick some colonies, you'd send out sequencing eventually, and then when you look at your sequencing result, you'd know for sure if you'd created the correct construct. Okay, we're going to talk about that process in some depth in part 3 of today's webinar. Okay, so come back in 20-30 minutes and we'll be talking about that. But an important note, I really want to highlight this for transformations. Controls for transformations are really important, especially negative control or some sort of vector-only control. That's really important. And the reason why is you want to make sure you know how many colonies should be appearing if your transformation failed, right? So if you see a whole lot of colonies on your negative control, your actual experiment with a lot of colonies probably didn't work, and it's not worth your time to go through all those steps for the quality control. If you have a vector-only control and it shows 5 or 10 colonies, and then your experimental transformation shows 50 colonies, oh, that's okay. Yeah, a few of those 50 colonies might be negatives, they might be false positives, but most of them are probably good, and so that's okay. And so it's really important to have these negative controls. It's also great to have a positive control to tell you if your transformation technique worked, but the negative controls for transformations are super important, all right? Now, earlier I gave you that link to the Fisher website. There's also protocol.io. This has lots of protocols on it. If you were to search for bacterial transformation, you'd find some protocols there that might be more applicable for what you're doing. I regularly use this protocol for transformations from the Barrick lab, and this also contains a procedure for making chemically competent cells. The reason why I like chemically competent cells sometimes is that they're a lot cheaper. So if you do electroporation, you need that device, the electroporator, and you need electroporation cuvettes, which can be pricey. Chemically competent cells, generally, the cost is up front making the cells, and you can usually make that pretty low. And then everything else afterwards is just simple plastics. So just something to consider when you're considering which type of competent cell to use or to make. All right. Okay, so questions. Hopefully from this part of the webinar, you guys learned about transformation some. You saw some of the important steps in the process. I gave you a lot of links here, and there's a lot more online, so you can always find more information about that there. But hopefully this is going to be a good start. If you're looking to do some transformations, it'll help you think about, you know, what antibiotic you're using, which is the selectable marker, what cell lines you might be using. Are you going to do electroporations? Are you going to do chemical transformations? How are you going to make your competent cells? Quality control is important. Okay. So talked about a lot of things there. Again, I'm sorry for that mess up at the beginning. I didn't realize the notes were being shared instead. So let's go ahead and stop the actual presentation there and see if there are questions. Happy to answer any question or comments. Just let me know, guys. It always takes a little bit of time for people to feel comfortable asking questions. Have people transformed much before? You guys can write this in the chat if you want. Have people done transformations much? As a rule of thumb, only one plasmid per cell. I would say, I would say generally we can say yes, but a lot of the mechanism behind how plasmids get into the cells during transformation, it's not fully clear. And this is something that every time I teach this, I double check and look to see if anything's been updated. And basically people have ideas about what goes on there in terms of pores opening up or little temporary pores being made, but it's not really clear to everybody. Generally, you just transform one plasmid at a time, but you can do two. It's definitely something I've seen people do, but generally you just do one plasmid at a time. Okay. So there's a question here from Aparna. What can be the industrial application of transformation at very large scale? Well, if you're going to, let's say you're in industry and you want to create some new product, right? You got to genetically modify your organism. So you can either engineer the chromosome, or you could make a plasmid and then transform that plasmid into your cells. So I think that's one possibility. If you're talking about doing lots of plasmids at the same time and multiplexing, you could do that as well, but you need to have something that allows you to differentiate one plasmid from another. So if plasmid number 1 gets into a cell versus plasmid number 2, how do you potentially differentiate those? And generally, people don't do that too much. So, let's see here. Michael asks, sort of plasmid from one origin. Could you specify a little bit more what you mean by plasmid from one origin? I mean, there are many different origins that exist naturally for plasmids. And if you're going to transform multiple plasmids in, they do need to be of different origins. So if you have one origin family here and a different origin family here, those two plasmids can be compatible within the same cell. But if the origins are the same or too similar and you put both plasmids into the same cell, you can have conflict and that'll be less successful. So yeah, yeah. So yeah, that's right, Michael. Yeah. So one origin family per cell. Is electroporation helpful? I don't know of it leading to damage. I think electroporation is perfectly fine. I've never seen anything that tells me that it's more damaging or mutating toward the DNA. What I'll tell you is generally people think electroporation is more efficient in transforming, but I've seen some chemically competent cells that are just as good. So I think it's really just a preference of technique. It can be more useful in some ways and more efficient, but it is more costly. So for example, if I wanted my students to do 50 transformations, that would be just in electroporation cuvettes, that would be potentially $100 or something like that, because you have to buy these plastic devices which can be reused but are more difficult to work with. Whereas competent cells, once you make a bunch, you make a bunch. Yeah, so yeah, what is better, transformation done chemically or electroporation, better results? Again, generally people think electroporation is more efficient, but there are definitely some cases where chemically competent cells work almost as well. So I would say you sort of have to sort of judge between efficiency and cost. And the cost to me is the really big difference between the two methodologies. Maybe 20, 30 years ago, electroporation was clearly better and chemically competent were not quite as good. But nowadays, a lot of companies have changed up their how do you make competent cell procedures. They've changed this up a lot so that you can acquire protocols or cells that are very competent and work really well. That plastic sentence. Oh, so electroporation cuvettes, yeah. So electroporation cuvettes are really pricey. They're like a couple bucks each. And so you'll often use them and then some people just throw them away. But you can actually find ways to recycle or reuse electroporation cuvettes. You have to do a very thorough cleaning, which involves soap, water, ethanol, 70% ethanol. If you go through that cleaning process, you can often reuse them. Some people will also put them under UV light and let them sit under UV light for like a day or something to kill anything in there. And that can be a very effective way to reduce cost. Here's the problem. They will eventually basically break down and decay, degrade from all of their uses. So you can usually get a few uses out of them, but eventually you will have to toss them. I would tell you this: if you're going to try it, try to use it with things that don't really matter, because you don't want to accidentally cross-contaminate anything. But you can definitely reuse the cuvettes. You just need to have a nice procedure for it. If you really want a procedure, email me and I'll try to dig one up. We have one from a couple years back. Okay. All right. I'm going to say that's it for question times here, guys. If you have more questions, as always, you can reach out to us via email, which is at the bottom of the page here. Sorry, somebody asked about the recovery. If I didn't mention this earlier, what you do is you let them grow at their normal temperature, let's say 37 degrees Celsius, in a rich media, so LB or more likely SOC. And what you do is you just let them grow and shake for like an hour without any antibiotics. And during that time, the genes on the plasmid will start to be expressed. And then after that hour's up, you can take the cells and put them on a plate and let them grow and incubate at the normal temperatures. So the temperature for that recovery time, again, about 37 degrees if you're going to work with E. coli. All right, not room temperature, and definitely not a fridge. Okay, hopefully that answered that last question. Okay.